Saya izin memulai acara. Ya. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. His Excellency Professor Hiroshi Suzuki from Obihiro University of Agriculture and Veterinary Medicine, Japan. So, His Excellency, the Dean of Faculty of Mathematics and Natural Sciences, Mr. Dr. Andes Abdul Ghafur, MSc, MSc, PhD, Honorable Moderator, guests, and all of the audience. Welcome to guest lecture 4th of December 2020 at the Department of Biology, Faculty of Mathematics and Natural Sciences, Tambung Mangkurat University. My name is Gustinur Aida Fasha, or for short, Aida. Please allow me to be your master of ceremony in today's agendas. So first of all, let we praise to our God who has given us mercy and blessing so we could attend this event today of guest lecture for basic and applied techniques of reproductive biotechnology presented by our honorable guest speaker, Professor Hiroshi Suzuki. Then in this very special occasion, we have several agendas, so allow me to read several segments of our agenda today. The first is opening, so the second is delivering rules and procedural of this event. And the third is listening to Indonesia Raya song. So the fourth is reciting prayer. The fifth is opening speech by the Dean of Faculty of Mathematics and Natural Sciences. The sixth is photo session. And then the seventh is our main agenda today. It is the speech and lecture of basic and applied technique of reproductive biotechnology presented by our honorable guest speaker, Professor Hiroshi Suzuki from Obihiro University of Agriculture and Veterinary Medicine, Japan. The eighth is discussion session, and then the last is closing. Well, then let's open our event today by reciting Basmalah. Bismillahirrahmanirrahim. Okay, so dear audiences, I would like to deliver the rules and procedural of this event, so please listen very carefully. The first is participants are expected to join the Zoom meeting at 9:45. And the second, participants are expected to use the virtual background that has been given by the committee. And if the participants have successfully joined the meeting, they have to first deactivate the microphone by clicking the speaker icon until it is mute. So deactivate the video by clicking the camera icon until it turns off, and fill in the attendance list in the link which will be shared in the chat room by the committee. So activate the video feature during the photo session. And fourth, questions related to the material are submitted through the Q&A feature or chat room with this following format: name, underscore student ID, underscore the question. So for better understanding, I would like to deliver it in Indonesia too. Jadi ada beberapa peraturan yang harus kita taati bersama. Jadi yang pertama, itu peserta diharapkan sudah join dalam Zoom meeting pada pukul 9.45 waktu Indonesia Tengah. Kemudian yang kedua, peserta diharapkan menggunakan virtual background yang sudah diberikan oleh panitia. Lalu apabila sudah berhasil masuk ke dalam Zoom meeting, peserta wajib yang pertama menonaktifkan mikrofon, kemudian juga menonaktifkan video, lalu mengisi daftar hadir di link yang akan di-share melalui fitur chat, kemudian mengaktifkan fitur video saat sesi foto. Dan peraturan yang terakhir itu yang pertanyaan terkait materi nanti disampaikan melalui fitur Q&A atau pada chat room dengan format nama, kemudian underscore, lalu NIM, lalu underscore pertanyaan. Nanti untuk peraturan ini juga akan kami share melalui chat room. Well, then the next agenda is listening to Indonesia Raya song. So for all of the audience, please listen very solemnly. Jadi acara selanjutnya itu adalah mendengarkan Indonesia Raya. Mohon didengarkan dengan hikmat. Okay, thank you for all of the audience. Terima kasih kepada para peserta. Step on to the following agenda, which is reciting prayer. Hope this event will be carried out well without any obstacle in it. So, let we pray based on our individual belief. Agenda selanjutnya adalah berdoa sesuai kepercayaan masing-masing. Silakan berdoa sesuai kepercayaan masing-masing. Berdoa dimulai. Berdoa selesai. So the next agenda will be opening speech by the Dean of Faculty of Mathematics and Natural Sciences, Mr. Dr. Andes Abdul Ghafur, MSc, PhD. Please, sir, time is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wassalatu wassalamu ala rasulillah. The Honorable Professor Hiroshi Suzuki from Obihiro University of Agriculture and Veterinary Medicine, Japan, the head of the Department of Study Program of Biology, Faculty of Mathematics and Natural Sciences, Universitas Lambung Mangkurat, faculties and students of Biology Study Program, and everyone who has joined this guest lecture. Ladies and gentlemen, as we have all been well informed that this morning we are going to have a guest lecture from Professor Suzuki, and the topic is about reproductive biotechnology. And biology itself is a broad medicine. It covers objects from molecular to organismal and up to ecosystem levels. And its branches vary from classical studies such as anatomy and taxonomy to the highly sophisticated molecular biology and biotechnology. And biology is also multidisciplinary. Biologists can and has benefited from physics, such as in microscopy and other instrumentation, from chemistry in methods of chemical analysis, from mathematics and statistics in modeling and data analysis, and even from computer science. So nowadays, biology, no biologist can be a true biologist who knows everything about biology. So experts are specialists on certain subdivision, but with in-depth knowledge and experience. Having said that, students of biology, and especially in undergraduate education in Indonesia at least, we need to have a good insight of all part of biology. So they have a strong foundation for specialization, for collaboration, and for the creativity in future career as biologists. The critical thing is that with the new discoveries, things become more and more complex, and our standard in learning different topics become higher and higher. For example, let's say 40 or 50 years ago, undergraduate students in genetics would learn only Mendelian inheritance, but now it's not enough. Similarly, students of animal physiology learn natural ways of fertilization, but now it is not enough anymore. They have to learn more things, but with more or less the same amount of time. So the question is how? How is that possible? So here's the trick. The key is learn from an expert, because experts can make the complex simple. So ladies and gentlemen, I am myself a biologist, and in my undergraduate study, I learned about reproduction and fertilization, but I know nothing about reproductive biotechnology. So we are lucky today to have Professor Suzuki with us to explain the basic and applied techniques in reproductive biotechnology. I'm very sure that he knows the essence and can make the complex things simple to digest for us. So students, I do hope that you don't miss anything during this lecture. And to Professor Suzuki, on behalf of all staff of Faculty of Mathematics and Natural Sciences, I thank you very much for your commitment to give this guest lecture. I hope that this is not going to be the first and the last. We are looking forward to having your contribution to our students and staff in the future. So ladies and gentlemen, please allow me to officially open this guest lecture by asking you to recite together Basmalah. Bismillahirrahmanirrahim. I wish you a happy webinar and a happy learning. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, sir, for the great speech. Okay. The sixth agenda is photo session, so please turn on the camera so we could all include in the frame. Jadi kepada partisipan, kepada audiens, silakan dinyalakan kameranya. Kita akan memasuki sesi foto bersama. Ya, kami beri waktu untuk menyalakan kameranya, untuk mempersiapkan kamera. Persilakan. Mungkin we could take several times of photo. So I will count to 3. Please smile at the camera. Jadi kita akan mengambil beberapa sesi foto. Jadi tolong tetap lihat ke kamera dan kita akan mengambil sesi foto. Jadi saya akan menghitung sampai 3. Jadi untuk yang pertama, 1, 2, 3, please smile at the camera. Okay, so the second, 1, 2, 3. Again, 1, 2, 3. Ya, terima kasih atas partisipasinya kawan-kawan dan Bapak Ibu sekalian. Then we come to the next agenda, which is our main agenda for today. The seventh agenda is speech and lecture delivered by Professor Hiroshi Suzuki from Obihiro University of Agriculture and Veterinary Medicine, Japan. Then for this next agenda, which is our main agenda for today, it will be guided by our beloved teacher, our honorable lecturer, Mrs. Ani Nurliani, SSE MSc PhD. She will be our moderator for the main agenda and our discussion today. So for Mrs. Ani, please, time is yours.
Thank you very much, Aida. Good morning, ladies and gentlemen. Hopefully, all of you are always in healthy condition. Thank you very much to Dean of Faculty of Mathematics and Natural Sciences, Dr. Abdul Ghafur, for your attendance and the opening remarks in this guy's lecture. Thank you very much also to Head of Department of Biology, Dr. Gunawan. Thank you for all of faculty members and all of students who already joined today in this guy's lecture. My special gratitude for Professor Suzuki. Good morning, Professor Suzuki. Welcome to Lambung Mangkurat University. Thank you very much for your willingness to deliver lecture at our Department of Biology, Faculty of Mathematics and Natural Sciences. Professor Suzuki, how about the current situation in Obihiro now?
So now it's become colder, so they're getting winter. Yeah, but you know, we don't have that not so serious problem regarding the coronavirus infection. Yeah, almost they're safe.
It's a good news. In Indonesia now, still there are many cases here. The current report mentioned that there are more than 500,000 cases in Indonesia now. So we hope the situation will be getting better soon and return to be normal like before.
Yeah.
Today, there are about more than 80 participants who join in this lecture. For the background of the participants here, there are several faculty members from Department of Biology, Dr. Rosmiati, and others faculty members. And there are some students coming from the Science and Animal Technology class and the other one coming from Microtechnic class. I believe they are very excited to hear lecture from you because this is a valuable opportunity for us to join in your lecture. Okay, ladies and gentlemen, before we come to the main session, let me deliver a brief biography about Professor Suzuki. Currently, Professor Suzuki is a professor at Research Unit for Functional Genomics, National Research Center for Protozoan Disease, Obihiro University of Agriculture and Veterinary Medicine. Professor Suzuki received bachelor degree in agriculture from School of Veterinary Medicine and Animal Science, Kitasato University in 1983. From the same university, he received master degree in 1985 for a thesis entitled Studies on the Development of Mouse Embryos Derived from Fertilization in Vitro. In 1995, he was awarded doctoral degree in veterinary medicine from University of Tokyo for a thesis entitled Studies on the Development of Zona-Free Mouse Embryo. For academic societies, Professor Suzuki is active as Executive Director in Japanese Society for Ova Research. He is also a counselor for Japanese Society of Animal Reproduction and one of members of Society for the Study of Reproduction. For his research experience, Professor Suzuki has already published about 300 publications, 12,000 more citations with H-index 55. It's amazing for us because it's still difficult to publish in Indonesia. We hope we can follow your successful. He conducted research related to development biotechnology, such as cryopreservation of embryo, sperm, and ovary. His current research is about cryopreservation for canine spermatozoa to be applied for the improved breeding of the guide dogs for the blind people. Professor Suzuki is also interested to protozoan infection. He focused on host parasite interaction with modified physiological condition of host animals using transgenic technology. That is a short introduction about Professor Suzuki. For today, Professor Suzuki would like to deliver lecture about basic and applied techniques of reproductive biotechnology. And without further ado, let's start the lecture session. Professor Suzuki, I'm giving the time to you. Now, the time is yours.
Yeah, thank you very much for your introduction. And also thank you very much giving me the opportunity. It's my great pleasure to give a lecture today. So then today, can you see the slide? It's okay?
Yes, yes, everything works fine, Sensei.
Okay, so then first of all, I'd like to remind you that what is the reproductive biotechnology. Reproductive biotechnology is a science and a technology that artificially modify the reproductive process to produce offspring in animals. So then, and also I'd like to remind you the pre-implantation development of mouse embryos in vitro. So then you can see the pronuclear stage of the fertilized eggs. You can see the both male and female pronucleus here, and they extruded second polar body. After about the 18 hours after the insemination, this fertilized eggs develop to the two-cell stage. And the next day, you can see the four-cell stage of embryos. And after that, so then initial differentiation of the cells initiated at the before modular stage, so-called the compaction. So then inner cell of the morula differentiated to the inner cell mast cells, which differentiate to the fetuses. The outer alignment of the cells are differentiated to the extra embryonic tissues. So then after accumulation of the blastocyst, so the embryo developed to the blastocyst. So then recently we used the time-lapse incubator, which is equipped with the camera, taking a photo every 15 minutes. And after observation by using such a time-lapse system, we identified the after initiation of compaction. So then after you can see that temporary decompaction can see. So then after that, the morula would be completed. So this slide does show the overview of the pre-implantation development of the mouse embryos. So then in the olden days, it started with breeding and reproductive techniques such as artificial insemination and cryopreservation of semen that were put into the practical use in livestock. But since 1970s, IVF, in vitro fertilization, cryopreservation of embryos, and embryo transfer have been successful one after another in experimental animals and livestock. So then the reproductive biotechnology has become essential for the current life science. Based on above, in 1980s, a new research area called transgenic technology, transgenics, was created, which was linked to the genetic engineering and enabled the genetic manipulation of individual animals, so-called transgenic animals or knockout animals. On the other hand, it also plays a major roles as a basic technology that supports human-assisted reproductive technology. Art has created a new occupation called embryologist in infertility treatment in human. So this slide shows the techniques for the reproductive biotechnology. This technology consists of the culture of embryos, manipulation of egg and embryos, embryo transfer, and the preservation of genetic materials. So then today, I'd like to overview some of these techniques, including IVF, ICSI, intracytoplasmic sperm injection, and ovarian transplantation, cryopreservation of sperm and embryos. And also, I'd like to talk about the freeze drying of spermatozoa. So then this slide illustrates the procedure of IVF in mice. So then at first, we have to remove the testis and the epididymis. In mice IVF, we use the epididymal spermatozoa, not ejaculated spermatozoa. They cut off the cauda epididymis and they make a small incision at the tip of the cauda epididymis and scoop a drop of sperm and they immediately put into the medium and incubate for 1 to 2 hours to induce capacitation. As you know, the ejaculated or the sperm stored in the cauda epididymis cannot penetrate to the eggs. They have to get the so-called capacitation, which is the physiological and biochemical change in sperm. But that capacitation easy to induce when you incubate the 1 to 2 hours in the media. So then induce the capacitation induced sperm add to the medium containing ovulated eggs. So then super ovulated eggs were recovered from the oviduct. So then the oviduct is put into the paraffin oil. It's next to the culture media. Then ampulla of the oviduct is turned and the eggs surrounded with the cumulus cells were introduced into the medium. And then a small amount of the sperm suspension added to the medium. So then after insemination, so then fertilization occur within 1 hour. So then application of the in vitro fertilization for biomedical science, it include the breeding of reproduction of the transgenic mice and animal models for human diseases. And the drug safety evaluation, especially reproductive developmental toxicity test. And also IVF utilized for the mass production of animals. And by using the IVF, you can eliminate the specific pathogens from the infected animals. And also IVF and the cryopreservation is available for the transportation of the genetic resources instead of the transportation of the live animals. And also IVF are used to for the making the transgenic animals. So then this slide shows the reproductive performance from natural mating in some strain of mouse with different ages. So then one of the most important the subject is that age-related decline in fertility. So then when the virgin both virgin male and females are mated with each other at 2 months old, from 2 months old to 15 months old. So then, and the delivery rate, the almost 100% between the 3 to 5 months old mice. But delivery rate decline with aging. So then, and at about 6 months old, so then reproductive performance rapidly decline. So then resorption rate will increased. So then when you maintain the mouse colony, so after 5 or 6 months old, if you have to apply the in vitro fertilization for maintenance of the mouse colony. Then after 10 months of age, you cannot get any progeny. But if you use the IVF, you can get the baby even in these the couples suffering from the infertility with aging. So this slide shows the result of in vitro fertilization of 4CS mice in different ages. So then age of mice here are 2 months old to 11 months old. So then even after the 7 months old, you cannot get the progeny, but if you use the IVF, so then you can get the even small amount of the small number of fertilized eggs from the such infertile mice. So then when you transfer the embryos derived from in vitro fertilization, you can get the newborns even in from the 11 months old of animals. So then age-related decline in fertility can see even and also that in a human. So you know the multiparous women and keep the higher pregnancy rate with aging, but in the non-parous women show the rapid reduction in their fertility after 36 years old. So then, if you want to obtain the baby, and you have to try to make a baby before 36 years old. After that, we have to apply the IVF for getting the baby in the humans. So then this slide showed the mouse embryos fertilized in vitro obtained from 11-month-old females. So then, of course, 11-month-old females showed infertility in natural mating, but the IVF can create the beautiful the blastocyst in vitro. So then, when you look at the uterine tissues, so then accumulation of with aging, so then the destruction of uterine gland is observed. So then in mice, the both egg and the uterus, the abnormal, show the abnormal the phenomenon. And also the IVF can use for the mass production of the animals. So then when you use the only one male and the 40 of the females, you can get the over 500 fertilized eggs at once. And when for about 450 embryos are transferred to the recipient animals, half of them develop to the newborns. So then these are about 200 the newborns the half brother. So then easy mass production make easy to experimental schedule. So you can get a large amount of animals at once. So also we apply the IVF for the infertility treatment on the disease model. This strain it's the C57 black KSJ DVM, showing the obese diabetes. So then they suffering from the infertility. So but the female mice can ovulate the oocyte by using the hormone treatment. So then we applied the IVF for the both homozygous diabetic the mutant. You can get the normal fertilized eggs. So then usually when you maintain this strain of mice for breeding, you use the mating system between the heterozygous mutant. But if you apply the IVF, both the homozygous male and the females that are available for the breeding and the reproduction. Also, IVF is available for the elimination of the pathogen from mice. So then these strain of mice are infected with MHV, mouse hepatitis virus. And these two strains of mice are infected with the Pasteurella pneumotropica. So, but when we check the cauda epididymis cauda epididymis or sperm suspension or fertilization media, you cannot detect the pathogens no more. So then by using the in vitro fertilization procedure, you can eliminate the pathogen from the infected animals. Also, we apply this kind of technology for the transportation of genetic materials. When you put the cauda epididymis into the paraffin oil and keep the long time at 4 degrees centigrade refrigerator temperature, and use the those stored sperm for in vitro fertilization, they show the fertility with the normal fertilization rate. So then we apply the transportation of these kind of knockout animals by using the stored the epididymis. After transfer of the epididymis by using the truck, then we apply, utilize such stored epididymis for the IVF. So, and you can get the newborns and with a normal successful rate. So then for embryo transfer into the oviduct, so then you can see the ovary here and the oviduct here and the uterus here. So then you insert the micropipette to the fimbriae for injection of the fertilized eggs. So, and this is so then a little bit difficult to get the skill, but after the 3 or 4 times training, easy to do. I mean embryo transfer into the oviduct under the imparted stereo microscope. So then when you keep the mouse strain for 10 years, so as live animals, so then the huge amount of budget will be required. But if you use the cryopreservation of embryo or spermatozoa, so such cost would be reduced. So then we now we are trying to keep the embryo or sperm as a storage of the mouse strains, especially a transgenic strain of animals. So then significance of the cryopreservation of sperm or oocyte and embryos, recurrence from genetic mutation, measure to the accident, the simplification of transportation. It's easier than the whole body, and especially in export and import. So then, as I mentioned previously, the IVF and also the cryopreservation is available for freezing the infected animals and the prevention of infection. and the reduction in cost of breeding and reproduction, and the acceleration of breeding. So then nowadays, it's essential, the liquid nitrogen for making the transgenic animals, as well as maintenance of the mouse strains. So, but the problem would be appeared in the cryopreservation of the embryos. So then this slide shows that the estimated productivity of newborn from frozen sold mouse embryos. So then the cryopreservation consists of the several steps, such as collection of embryos, freezing, sewing, recovery of embryos, how much is the survival rate of the sold embryos, cultivation before the embryo transfer, so success rate of the embryo transfer. So then when you get 20 embryos from a female, so then successful rate is not 100% in each steps. So the number of animals, number of embryos reduced to finally, after embryo transfer, only 1 to 9 newborns would be obtained from 20 embryos collected. So then productivity is cryopreservation embryo is not high. Therefore, in order to preserve about 500 embryos in each strain of mice. So then colony to be closed must be expanded once. So that to avoid this contradiction, sperm preservation has come to be performed. Now, cryopreservation of genetic materials move from embryos to sperm. But fertility of cryopreserved mouse spermatozoa is quite low, especially C57 Black 6 strain of mice. These strains are most available for making the transgenic strain of animals. So then we have to overcome this difficulty. We apply the partial zona pellucida dissection. So then this PZD is a method for improving fertilization rate in the cases of severe male factor infertility, including oligospermia in mice. So then you cut the zona pellucida partially. So then sperm easy to access to the plasma membrane of the oocyte. So this is the procedure of the PZD, and you add 0.3 molar sucrose in PB1. You know, when you add the 0.3 molar sucrose, and the cytoplasm of the oocyte is shrunken, so then you can get the large space of the perivitelline space. So then you can cut a part of the zona pellucida by using the dental needle like this. So then after cutting the zona pellucida, you can recover the unfertilized oocyte with the dissection of the zona pellucida. So then this picture show the PGD oocyte after the insemination. So then you can see the female pronucleus here, second polar body here, and the slit of zona here. So, but this, the slit would be large. So the blastomere of the embryos escaped from the zona pellucida after the transfer into the oviduct. So then when you apply the PGD, you have to culture the embryos up to the morula or blastocyst stage to prevent such escape of the blastomere from the zona pellucida. So then we overcome to overcome this difficulty, we apply the micromanipulator system. So then we used the piezo micromanipulator. The piezo micromanipulator, when a piezo pulse is applied to the injection needle, it vibrates finely back and forth. The piezo micromanipulator is the ultra small rock drill. So then you can make an incision easier than the dental needle. So then when you use the piezo micromanipulator, you can control the legs of the slit easier than the partial zona PGD. So then if this the slit length is short, so the escape of the blastomere would be prevented. So then, as we expected, so then B, so this embryo received the zip with a small incision. Then after the IVF and the subsequent embryo transfer into the oviduct, so then you can see that normal development, embryonic development of such oocyte. So, but if slit was large, so majority of blastomere escaped from the zona pellucida. So then in conclusion, when you apply the dip to the unfertilized oocyte, you can transfer those fertilized eggs to the oviduct. So then we overcome the problem of the lower fertilization rate of the C57BRAC6 mice for the cryopreservation of the sperm. And also, so then this piezo micromanipulator also used for the sperm injection to the unfertilized eggs. So this is so-called ICSI, intracytoplasmic sperm injection. So then you can see the sperm head here. Then the plasma membrane of the oocyte is a very, very elastic. So then you cannot puncture the plasma membrane by pushing the injection needle. But when you apply the piezo pulse, So then you can insert the micropipette into the cytoplasm of the oocyte. So that you can see the sperm head here. And this is the micro injection system. This picture shows the micro injection of DNA into the male pronucleus. So then when you perform the ICSI, if you add the DNA fragment into the sperm suspension, so then the DNA fragment cooperated to the mouse genome during the fertilization. So then making the transgenic animals, for making the transgenic animals, we used the DNA microinjection into the fertilized eggs, but also we can utilize the ICSI system for making the transgenic animals. And also the piezo micromanipulator were used for making the chimeric embryos. This is the host embryos, host blastocyst. That you can see the embryonic stem cell here. Then when you apply the piezo pulse here, you can insert the micropipette containing ES cells. Then after injection of ES cells, you can make the so-called chimeric embryos. So then if the host embryos are derived from the mice colored white albino, and the ES cells are derived from the mice colored black, after transfer of such chimeric embryos, you can get such chimeric chimera. For this technique, it's available for making the knockout animals by using the embryonic stem cells. So then I'd like to remind you that what is the embryonic stem cells? What is stem cells? Stem cells are primary undifferentiated cells which retain the ability to differentiate into the other cell types. So then stem cells have the potential self-renewal and differentiation to the other cell types. So then this ability allows them to act as a repair system for the body. The medical researcher believe the stem cell research in the field of regenerative medicine has a potential to change the face of human disease by being used to repair specific tissue or to grow organs. So then they prefer to research the stem cells. So then we have to know there are several types of stem cell here. So then stem cells are categorized by the potency, which describe the specificity of that cells. So one of them is the totipotent stem cells. Totipotent stem cells are produced from the fusion of an egg and sperm cells. This means the fertilization. Cells are produced by the first few divisions. Cells mean equivalent to the blastomere. Blastomere produced by the first few divisions of fertilized eggs are also totipotent. So then these cells, totipotent cells, can grow into any type of cell without exception. The example of the totipotent stem cell, the zygote, early stage of embryos. Pluripotent stem cells are descendants of the totipotent cells and can grow into any cell types except for the totipotent stem cells. So then embryonic stem cell, ES cells, is one of the pluripotent stem cells. embryonic stem cell is not have not a totipotency. So then third one is the multipotent stem cells. These cells can produce only cells closely related to the family of cells.
Hello, Suzuki Sensei. Are you hear me?
Terputus Kak Bu Ani?
Ya, sepertinya ya.
Ya, kita tunggu sebentar.
Iya. Bagi mahasiswa yang ingin bertanya, bisa mulai dari sekarang menyiapkan pertanyaan. Jadi pertanyaan bisa ditulis dalam bahasa Indonesia, kemudian kami akan bantu untuk menerjemahkan. Atau mahasiswa juga bisa langsung bertanya dalam bahasa Inggris. Format pertanyaan itu adalah sebutkan nama, kemudian NIM, dan selanjutnya adalah pertanyaan.
Sorry, our system has been terminated.
Oh, okay, no problem, Sensei. You can continue.
Okay. So, can you see the slide?
Yes, we can see.
So then this slide show the human blastocyst. So then embryonic stem cell is derived from the inner cell cells, which differentiate to the fetuses. So then after establishment of the cell line as embryonic stem cells, so then when you put some growth factors into the medium, so these cells could be differentiated to any kind of the cell types. So, but you know, the ES cell has no totipotency. When you transfer the ES cell to the uterus, after ES cell transfer, you can see so many kind of the differentiated tissues into the uterus, but you cannot get the whole body. So embryonic stem cell lack the differentiation potential for the extra embryonic tissues. So then ES cell does not have the totipotency. But so then ES cell is derived from inner cell mass. So then the inner cell mass cells can differentiate to the germ cell, of course. So then if you put the mutation to this cell lineage, So then you can get the mutated germ cells. So we apply this system for making the knockout animals. So then these are the embryonic stem cell. So then you put the targeting vector to destroy the some gene of interest. So then you can induce the mutation of the gene of interest in the embryonic stem cells. So then when you make the chimeric embryos by using the ES cells with mutation, so then these mutated ES cells differentiate to the testis, I mean sperm producing tissues. Then the testis produce the sperm with mutation. So then this is an excellent phenomenon. When you put the mutation in the culturing cells, then you obtain, after the making the chimera, you can get the live animals with mutation by mating after getting the chimeric mice. So then there are many methods for the identification of the role of novel gene of interest in both in vitro and in vivo. But if you know the interest in the gene regarding the hypertension or diabetes, you cannot measure the blood pressure here, or you cannot measure the blood sugar level in vitro. So you need to examine and the gene function in vivo by using the knockout animals. So then we turn to the next subject, freeze drying of spermatozoa. So then cryopreservation of sperm has made a great contribution to reproductive biotechnology, including infertility treatment, preservation, and transportation of genetic materials. So then cryopreservation of mouse sperm using liquid nitrogen has been carried out in the large quantities, especially from the late 1980s, due to the development of gene knockout technique in mice. So then, however, if freeze drying makes it possible to store sperm at room temperature, like coffee, or in a refrigerator temperature for a long time, the maintenance cost will be greatly reduced. And further, freeze-dried sperm can be easily sent to in an envelope. It seems that there are great expectations for practical application of freeze-dried sperm.
Ya, kembali ya, ada sedikit gangguan penerimaan di sini untuk Suzuki Sensei. Mungkin karena jarak yang berjauhan ya, berbeda negara, jadi gangguan ini sangat dimungkinkan terjadi. Jadi kita tunggu. Mudah-mudahan tidak lama. Apakah sudah ada pertanyaan yang kira-kira akan diajukan dari mahasiswa?
Sorry, our system is not stable right now.
It's okay, Sensei. You can continue now.
And attempt of the freeze drying of spermatozoa has been identified from the 1950s. Some of the reports show that getting offspring or confirmation of the conception. But all of those reports could not be reproduced. So it might be difficult to gain the motility of spermatozoa after freeze drying. So then nowadays it's difficult to they gain the motility after freeze drying of the spermatozoa. So then ICSI
Ya, ini sudah ada satu pertanyaan ya dari MC memulai. Ya, jadi nanti kami siapkan pertanyaannya dalam bahasa Inggris. Mungkin yang lain bisa menyusul untuk mengajukan pertanyaan, dipersilahkan sambil menunggu Suzuki Sensei untuk connect kembali dengan kita.
Sorry.
Ya, silakan. Okay, no problem, Sensei. Now it's okay. You can continue your presentation.
Japanese system is not good.
It's okay. Sometimes it's happen when we have a long, far distance.
So I know so then Professor Yanagimachi described the comment in the review. So then spermatozoa that had been snap frozen without protectant, and those which were freeze dried were all dead in a conventional sense. But they are able to produce live offspring by ICSI. So then these findings raise an interesting question as to life and death of spermatozoa. Those spermatozoa with broken plasma membrane are obviously dead as cells, but they are able to produce normal offspring. In that sense, they must still be alive. So then after the development of the reproductive biotechnology, so then now definitions of life and the death of spermatozoa as thus arbitrary. So then, but is it possible to store freeze-dried sperm for a long time? With the development of the ICSI, freeze-dried sperm has recently attracted attention as a new method for long-term storage of sperm. From the viewpoint of the using freeze-dried sperm for preservation of the gene resources, although guaranteeing the possibility of long-term storage of freeze-dried sperm is an important issue. Possibility of long-term storage such as 10 or 100 years hasn't been examined, as its implementation is difficult. So then we attempt to apply the accelerated test using Arrhenius plot, which is generally used to consider the stability of pharmaceuticals in terms of kinetics to freeze-dried sperm and examine the possibility of the long-time storage of freeze-dried spermatozoa. So then after freeze-drying of the spermatozoa, we store those samples at 30, 40, 50 degrees centigrade for up to 7 days. Then after the storage, we applied those spermatozoa to ICSI and cultured up to for 96 hours after ICSI. This is the chart of the freeze drying. It's difficult to see. Then this slide illustrates the previous chart. This yellow line, the self temperature, black line is the sample temperature, vacuum pressure, primary vacuum, and the secondary vacuum pressure. And then green line, the ice condenser temperature here. So then we after the storage of the freeze-dried spermatozoa at 50 degrees centigrade for 1 to 7 days, the developmental rate of the embryos decreased with storage time. Even 40 degrees, 30 degrees, and also reduction in the developmental rate to the blastocyst stage. So then we plot the development to the related to cell stage embryos like this, and getting the Arrhenius plot here. So then this is the predicted value by Arrhenius plot. Now, if you store the freeze-dried sperm at 25 degrees centigrade, after 10 years, you cannot get the offspring no more. But when you put the such sperm at the refrigerator, 22% of the embryos develop to the two-cell stage. The much lower, minus 20, minus 80, is almost equivalent to the unstored controls. And the actual data here, the report almost equivalent our estimation. So then this is the development to the blastocyst stage. So then if you want to get the blastocyst after the 100 year, you have to keep the freeze-dried spermatozoa at -80. So this slide shows the result of the comet assay of freeze-dried spermatozoa. You can see the comet tail here. This means the destruction of the DNA. So then when you keep the -80, the comet tail does not appear like the fresh control. So then, in our estimation, so if you keep the samples below -70, so long-term storage would be available. But the below -80, it's difficult to keep the long time. For example, -20, it's available only for 4 years. So then summary of our accelerated test, storage at refrigerator temperature. So predicted developmental rate up to 2-cell stage was 22 after 10 years storage. And it was predicted the 2-cell stage embryo would not be obtained after 100 years of storage. So then storage at -80, even after storage for 100 years, development rate up to 2-cell stage was almost 100%. No decrease in developmental rate. Developmental rate to blast stage was not predicted to decrease even after the storage for the 100 years. So then it was suggested that the -80 or lower is suitable for long-term storage of freeze-dried sperm under the commonly used freeze-drying conditions. About vacuum pressure 0.04 millibar. So then we try to improve the storage stability of freeze-dried mouse spermatozoa. Then we changed the primary drying pressure. Then also accelerated test would be had been performed. So then I will show you that conclusion like this. So then if you decrease the vacuum pressure to the 0.37, so then development to the live fetus is improved. So then the primary vacuum pressure is the important factor for freeze drying. So then even in the freeze dried the sperm stored at 4 degrees centigrade for months, 6 months. If you apply the 0.37 millibar in the primary vacuum pressure, overall efficiency significantly increased as compared with the conventional conditions. So the degree of vacuum during the primary drying is one of the important conditions in order to enable long-term storage of freeze-dried sperm at a relatively higher temperature. So then we tried to apply the freeze-dried the mouse spermatozoa for transportation of genetic materials. So then we transferred the freeze-dried sperm from Japan to Biologi and returned to Japan. So range the 3,800 kilometers. So, this slide shows the changing of the sample temperature from labo to labo. So then the freeze-dried spermatozoa are transported by the truck to the airport. So they from Narita, Tokyo Narita to Belgium, and they staying for several days in Belgium and return to Narita and transported to the lab. And those transported sperm were applied for the ICSI. Then result, when freeze-dried spermatozoa are stored at 80 degrees centigrade for 2 years, offspring are obtained at 28, 8, and 1% respectively. The freeze-dried sperm produced at 0.37 millibar and stored at -80 are not easily affected by exposure, transportation to the environmental temperature for about 1 week, only 1 week, and retain sufficient fertilization and developmental ability. So then next, I would like to talk about the so then next I'd like to talk about the breeding of guide dog for the blind people. This is application of the reproductive biotechnology for breeding of dog. So then problem is although guide dog makes a remarkable contribution to the quality of life of the blind people around the world, many countries suffer from the acute shortage of guide dogs. So then even among the Labrador Retriever, only 30% of dogs that were trained ultimately work out as guide dog in Japan. So then it's difficult to predict which puppy will make excellent guide dogs. So then you need the training of the candidate. Can you hear me?
Yes, Sensei, I can hear you.
Okay. So then we have tried, the development of the assisted reproductive technique for guided egg for the blind people, artificial insemination, sexing, cryopreservation, ovary, and so on. I'd like to show you the one of these techniques. And first of all, I'd like to show you the cryopreservation of ovarian tissues. So then we try to cryopreserve the ovarian tissues in dog and try to do the xenografting. This mean to examine the viability of the frozen thawed ovarian tissues. So xenotransplantation into the ovarian bursa of immunodeficient mice was attempted. So then after anesthetizing the mice, incision was made in the back and the ovaries were pulled out. And the cut was made from the fat on the side of the ovary. To tissues, the dog ovarian tissues are inserted. And the 4 weeks after the transplantation, and the ovaries were removed from the mice and the subject to the HE staining. So then, the mouse ovary it was stained with stained with hematoxylin eosin. It's much higher intensity and darker than the ovary, canine ovary. So then this slide shows that after 4 weeks after the transplantation, you can see the dog ovarian tissues here, dog ovarian tissues here, and the dog ovarian tissues here. So then this data indicate the cryopreserved canine ovarian tissues keep their function. And also we try to apply the artificial insemination of the dog. So first of all, usually the conventional cryo extender of the semen is consists of the egg yolk. That's similar to bovine semen. So egg yolk based, the complete the extender is the complicated component. So then this advantage of the egg yolk extender, the varied viability depending on the egg yolk quality. And also the conventional method of cryopreservation of the dog require the long equilibration time. So then when outbreak of avian influenza occur, some country the we could not import we-
Yes,
we,
Sensei? Mhm.
okay,
Okay,
can
Sensei.
you hear me?
Yes, I can hear
Okay,
you. No problem.
okay, okay, okay. So then we try to modify the component of the cryoprotectant of the for the cryopreservation of canine spermatozoa. Then we apply the skim milk and the sugar instead of the egg yolk. Then we, this is the proportion of the motile spermatozoa after cryopreservation. This is the name of the, the dog, male dogs. And as you can see, the motility after sowing is almost equivalent. No difference between the egg yolk and the skim milk-based extender. But no.
Sorry, Sensei. Your
Yes.
screen not appear.
Okay.
Okay. Okay, now it's available.
It's okay. Okay. So then this slide shows the delivery rate after artificial insemination. Then when you use the egg yolk extender, if you want to get the higher delivery rate, you have to perform the artificial insemination on day 5 and 6. But if you use the skim milk extender, only one artificial insemination is available for getting the higher delivery rate. And also we tried to reduce the incubation time. Usually we use conventional method then our data indicate even 2 hours equilibration times or 1 hour equilibration times, it's enough for getting the good motility of spermatozoa after cryopreservation. So then now we use the 1 to 2 hours equilibration time for making the frozen spermatozoa. And also we try to seek the further improvement. So then I'd like to show you the data, recent data, effect of quercetin supplementation on the motility of canine spermatozoa after cryopreservation. Quercetin is the flavonoid contain the subbactic trees with freeze resistance. And it's quercetin known to be the strong antioxidant. So we try to add quercetin to the extender. So we supplement the quercetin to the second extender here. And after cryopreservation, we try to check the motility and perform the artificial insemination. So this slide shows the motility of spermatozoa after cryopreservation. So the blue line indicate the control, I mean without quercetin supplementation. Both 5 microgram and 10 microgram quercetin supplementation improve the motility of cryopreserved spermatozoa. Then we apply the artificial insemination. So then this you know, the ID of the recipient female. We perform the AI day 4 after LH surge, day 4 and day 5 after LH surge. We observed in total 24 follicles by echo examination, and we obtained 18 puppies after the artificial insemination. So then the litter size of the this artificial insemination would be the 6.0. It's a quite enough number of puppies per females. Usually the litter size of the laboratory liver in 6.5 in natural mating. And this data is almost equivalent to the natural mating.
Okay, Sensei, you can hear me?
Okay.
You can continue, yes.
Okay. So in conclusion, we need to much more than female than male. So then we applied the flow cytometric sperm sorting method. This is the already launched for the cattle artificial insemination. So that you can you can buy the only X sperm or only Y frozen sperm. It's available. We use the bovine system for separation of XY sperm in dog. And we developed the detection X and Y chromosome on canine spermatozoa by dual color fish. So we have developed the fish method for the detection of XY chromosome. Then we confirm the sex XY ratio after sorting of the spermatozoa. So dog A, X fraction, sorting X fraction contain over 90% of the X sperm. Dog B, X fraction contain around 90% of X sperm. after conformation of the fish. So then when you use the X fraction of spermatozoa, you can get much more number of female dog after insemination. And also we have tried the embryo or cryopreservation. So then this is the flushing the uterus for getting the embryos. This is flushing the oviduct for getting the earlier stage of embryos. So then we have tried to freeze the canine embryos by vitrification method by using the ethylene glycol. Then after embryo transfer, non-surgical method, it's similar to the artificial insemination. Then we have succeeded puppies derived from liquid nitrogen. So then as I described, we have achieved the development of the new semen extender and the practical application of the artificial insemination in dog. And for the first time in the world, we have succeeded in obtaining offspring derived from frozen embryos by non-surgical embryo transfer. So we have time is up, right? No, I have to stop my talk. Thank you very much. I have to apologize the several times termination of the remote. Sorry. Thank you very much.
Thank you very much, Suzuki Sensei, for your presentation. It's a very interesting and insightful presentation for us. Okay, now we open the Q&A session. So there are there are several questions here. So I will read the first question from Aida. Okay. Oh, already translated by computer. Thank you very much. The first question is in embryo manipulation or in embryo transfer, is there a possibility of failure in cell division? So this is the first question, Sensei. Could you answer this question now?
Actually, no, but if you have the if you have the suitable culture condition, you can continue the normal development after manipulation of the embryos. So then we have to keep the temperature around 37 degrees centigrade in the lab, and the humidity and the atmosphere. So if you keep the condition like in vivo, cell division, you can expect the normal cell division, normal development of the embryos after manipulation.
So it's possible to to do embryo transfer in Indonesia, I think.
Yeah, it's possible. No problem.
Yeah.
No.
Okay, thank you, Sensei. For the next question, please, Komita, could you show the next question? Okay, the next question is, what kind of dogs are bred and trained to use as guide dogs for blind people? Are there any tips and tricks to increase the fertilization capacity of frozen mouse sperm in vitro fertilization technique? Okay, Sensei, it seems there are two questions from
Yeah.
Rani.
For guide dog for the blind people, and most of breed, most guide dog would be the from the Labrador Retriever or Golden Retriever. But some of association use the standard poodle. But the majority of the guide dog are derived from the Labrador Retriever and the Golden Retriever, and they are F1 hybrid.
So it's not for common dog. It's a special only for a specific dog, you mean?
No, they are soft and clever, and yeah. So easy to handle, easy to control, easy to train.
I see.
Yeah. Yeah. But any breed, large breed is available for the guide dog for the blind people. Yeah.
Okay. Okay, thank you, Sensei. For the second question, are you already read? Okay. Still, I think from the second question, we still have one more. Yeah, this one. Are there any tips and tricks to increase the fertilization capacity of frozen mouse sperm in vitro fertilization technique?
Yeah, as I mentioned, when they bypass the donor pellucidar, we can obtain the higher fertilization rate, even if you use the frozen mouse spermatozoa. But recently, Dr. Nakagata of the university developed the new culture medium for IVF using the frozen spermatozoa. So if you use such medium, you don't need to cut the donor pellucida before the insemination.
Okay. Okay, thank you. For the next question, we still have several questions. Okay, from Muhammad Erwansa. When doing research on embryos in mice, did you experience any problems in its implementation, and how do you anticipate it? So
it's difficult to answer. I cannot understand the actual meaning of the question.
Okay, maybe what is the big challenge, Sensei, in your research in this field?
What is the big challenge?
Yes. Maybe we can learn from your experience. So when we apply this technique in Indonesia, we can consider it.
Mm, mm. If when you use the mice,
Mm-hmm.
we don't have the problem. It's easy to do the research by using the mice.
Mm-hmm.
Yeah, even if you don't have the SPF facility,
Mm-hmm.
no problem. You can try to do the research by using the mouse. So I don't think that they are. I don't think that the rising problem. No, no, you can do it.
We can do it. Yes, we hope so, Sensei. Okay, the next question, please, Komita. Okay, from Sofa Maulida. Fertilization success rate with dried sperm is influenced by time and temperature of storage. Are there other factors that influence the success of fertilization?
Yeah,
Despite time and
the method of the freeze drying is not well developed. So we have to, modify, improve the freeze drying procedure, especially the vacuum pressure. So then moisture also it might be important factor is the moisture content of dried spermatozoa. So, but it's difficult. If at least in my lab, it's difficult to analyze the in the further experiment. Yeah.
So which one better, Sensei? Frozen spermatozoa or freeze-dried spermatozoa? Which one better?
Maybe you don't need if you freeze drying, you don't need the liquid nitrogen no more. And also now recently, freeze dried spermatozoa transport to the the space.
Oh.
Yeah, because of it's easy to the transport without liquid nitrogen. So in the NASA in the US try to transport the frozen freeze drying spermatozoa by using the rocket to the space. Yeah, this is only experiment. Yeah.
I see. Okay.
Yeah.
So we hope it will improve in the future about this technique. Okay.
Yeah.
For the next question, is still there are several questions. Please, Komita, can you share to us? Oh, okay. Maybe for now, it's enough for the question. I think no more question for you, Sensei. So,
Okay, thank you.
Thank you very much for your well explanation, Suzuki Sensei. It's a very interesting lecture, and this lecture provide many new insights for us about techniques of reproductive biotechnology. Someday we hope we can do in our lab. Yeah, maybe it's supported by you. We hope someday we can do research collaboration with you.
Okay.
Okay,
Okay.
thank you. So now this is the end of Q&A session. It's very nice to discuss with you, Sensei. Actually, I would like to give more time for discussion, but the time goes to the end. So I hope all participants can get beneficial from this lecture. I hope this lecture can motivate us to always learn many things because there is no limit to learning. And once again, I want to express my gratitude to Professor Suzuki for spending time in this guy's lecture. We hope we can invite you again in the future and do some research collaboration with you. We also hope this pandemic situation will be getting better soon. So someday you can visit our department to meet us directly and go around our hometown, Banjarmasin City.
Okay, thank you very much. Okay,
Thank you
I hope
very
so.
much. In
Thank
this
you.
opportunity, I also do apologize if there is anything in this event that is not like your expectations, Sensei. I'm so sorry. So, and I hope, yeah, this is not the last opportunity to meet you. I look forward to see you again in the future. Okay,
Okay,
thank
thank
you
you.
very
Thank you.
much.
Yes, Bapak.
Thank you very
See you.
much for our participants who already joined us from beginning until the end. Our Dean, Dr. Abdul Ghafur, Dr. Gunawan, all faculty members, and all students. Thank you very much. We really appreciate your participation. Thank you. I will end my session and give the microphone back to Master Ceremony. Thank you. Aida, please. Yeah, Aida, I end my session. Could you continue this guy's lecture agenda?
Thank you, Mrs. Ani, for guidance today. And of course, I would like to thank to Professor Hiroshi Suzuki for the amazing opportunity to learn something new today. So thank you, sir. And well, then the last agenda is closing. Let's close our event today by reciting Hamdalah. Alhamdulillahirabbil alamin. Finally, from the deep of my heart, I do apologize for my mistake while in presenting this event today. So, saya sebagai pembawa acara mohon maaf yang sebesar-besarnya apabila ada kesalahan dalam penyampaian. Dan thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Terima
wabarakatuh.
kasih Bapak, Ibu, dan kawan-kawan semuanya.
Thank you, Suzuki Sensei, and all student and faculty members.
Thank you very much.
Thank you very much. Ya,
Mungkin dipersilakan juga kepada kawan-kawan yang belum mengisi daftar hadir, silahkan diisi daftar hadirnya. Daftar hadir telah kami bagikan di room chat. Berarti yang peserta mikroteknik sama sains teknologi hewan harus mengisi daftar hadir berarti ya Bu?
Iya, wajib ya. Nanti ini akan direkap dan diikutkan di daftar hadir mahasiswa perkuliahan secara umum.
Ya.
Jangan lupa ya.
Jadi kepada kawan-kawan yang Yang belum mengisi daftar hadir, silakan isi daftar hadir dulu.
Terima kasih Bu Rani, Pak Gun, dan dosen-dosen yang lain. Ada Bu Asti, tadi ada Bu Rus, saya lihat. Mohon maaf yang tidak bisa disebutkan satu persatu. Terima kasih atas bantuan dan kerjasamanya hari ini.
Sama-sama Bu Ani. Aida, terima kasih.
Sama-sama
Aida.
Bu Ani.
Ya, terima kasih.
Terima kasih, Ibu.
Ya, ada Bu Rahmah kelihatannya ya. Saya tidak bisa mencek satu persatu tadi. Ya, semoga bermanfaat kuliah tamu kita pada hari ini.
Amin. Banyak yang bisa bahasa Jepang, Bu. Arigatou gozaimasu, Sensei Ani, katanya, Bu. Kemudian ada juga ganbate, katanya, kata Rahma.
Ya, ini Bu Rahma ini mencoba berbahasa Jepang. Nanti Bu Rahma coba ini
Ini
di bahasa Jepangi senseinya. Tadi pertanyaan ada 2 ya dari mahasiswa ya, Muhammad Erwansah dan Sopa Maulida.
Ibu Rahma, mohon maaf, Bu. Saya kira kawan-kawan mahasiswa.
Oke, saya izin live ya. Terima kasih banyak semuanya.
Terima kasih banyak.
Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Saya izin end meetingnya ya, soalnya converting recordnya lumayan lama. Boleh ya? Oke.
Terima kasih, Bu.
Terima kasih Aida.
Terima kasih, Bu, juga.
Oke.